Today we are going to look at viruses in a new way. We're going to see how studies of viruses has helped us to learn something about the nature of life itself, about the very heart of the living systems. Now, some scientists claim that viruses are only partly alive because they can be obtained in crystalline form and so forth. And you may wonder how studies of something that is perhaps only partly alive, has only certain properties of living things, can help us in the study of life itself. Well, to do so, we'd like to compare a virus with a living cell. Now, here is a model of a living cell enlarged about a 250,000 times. You can see all of the intricate machinery inside that cell. Now, these living cells come in a variety of sizes and shapes. Some are very long, slender things. This is actually a fairly small cell. Now, a virus such as poliomyelitis virus, to the same scale as this cell, would appear something like this. Now, viruses are peculiar in that outside they are inert, but when placed within certain living cells, they can spring into action and multiply many, many hundreds and thousands of folds over. If this virus were placed inside a living cell such as this, it's able to divert all of that machinery that you see in there and make many, many more of these little virus particles. Another peculiarity of viruses is that some of them can be obtained in crystalline form. And if you would slice a crystal of poliomyelitis virus and look at it by means of the electron microscope, this is what you would see. There's another class of viruses, the bacterial viruses. And here on the floor we have a drawing of one of these viruses that attacks germs. And still a third great class of viruses is the plant viruses. Here's a model of tobacco mosaic virus enlarged to about 65 million times. This cell that we just looked at, if enlarged to the same extent, would be about 1/3 of a mile across, and my head would be as large as the Earth. Well, now you may wonder how we can learn about the nature of life through studies on these seemingly only partly alive viruses. Now, this is a question that I'd like to put to my colleague, Dr. C. Arthur Knight, whose research in recent years has focused on certain of these lifelike properties of viruses.
Viruses are almost always thought of as disease agents, which of course they are. But how are viruses going to teach us anything about the vital functions of life? In order to answer this question, we have to consider briefly what life is. What are the major characteristics of living matter? This chart summarizes the major qualities of living things: organization, movement, irritability, growth, and reproduction. First, organization. Every living thing has a characteristic size and shape, which means that they organize their material in a very definite way. Usually the unit of structure of living things is the cell, like this model we have here. You can see that this is amazingly complex, and there are billions of cells of this sort in a plant or in an animal. Not all cells are round like our model. Cells come in a variety of shapes, as you can see in this drawing. But all cells have this common quality of organizing their material in a definite pattern. Movement is another quality of living things. We all recognize that animals move around a good deal, and plants do too. And we can see this if we speed up time. Even a plant in a quiet room will engage in a great deal of movement and activity. Likewise, even the insides of cells are in constant motion. Another quality of living things is irritability, which means that living things respond to their environment. They respond to stimuli. This scavenger cell is responding to a bit of dead cell in its vicinity, which it engulfs. An interesting example of irritability with a plant is this sensitive mimosa plant, which responds to the slightest touch. I think you can see that living things do indeed respond to their environment. Growth is another important quality of living things. Living things grow in two ways: by increase in volume of the cells and by increase in the number of cells. Both of these processes are nicely illustrated by the development of the human embryo. We can see in this picture the growth of the human embryo from about 19 to 120 days. Reproduction is the final and probably the most important quality that living things have. When a single-celled organism divides, a new individual is produced. But with the many-celled organisms, a new individual is produced only by the union of two special cells, the sperm and the egg. The fertilized egg then proceeds to divide, as this picture of a salamander egg shows us, and division continues until a stable form is reached. It is hard to overemphasize the importance of reproduction. Without reproduction, life on Earth would cease to exist, and the Earth would become as barren as the moon. This is easily the most important quality of living things. Now we can draw some conclusions. First of all, living things have these qualities that we've listed on our chart. But what about non-living things, and where do viruses fit into the picture? Some non-living things may have some of these first four qualities to a limited degree. A crystal may, for example, grow and have some organization. But non-living things never reproduce. Turning to the viruses, they may also have some of these first four qualities, but they also are able to reproduce. So we see that viruses truly lie in a shadowy region between living and lifeless objects, possessing some of the qualities of both. To understand more clearly how viruses may help us to understand this vital process of reproduction, we need to turn again to the cell and look in more detail to this process. We find the key to reproduction is in the heart of the cell, in the part which we call the nucleus, which you see here in the center of our cell model. Biologists have discovered some very important bodies, the chromosomes, in the nucleus, and they appear as fuzzy bands in our model. We also have here a model of a single chromosome, as it might look if it were magnified many thousand times. Now, when the cell is about to divide, the chromosomes duplicate themselves, and then as the cell divides, the chromosome pairs are pulled apart, and one set goes to one cell and the other to the other cell, so that each cell ends up with a complete complement of chromosomes which are usually exactly alike. This process of separation of the chromosomes is illustrated in our drawing, which shows the chromosomes as they are pulled apart. Next, let's look at this process as it actually occurs in a living cell. You see again the chromosomes getting arranged in the center of the nucleus, and then the pairs are splitting and chromosomes are being drawn in opposite directions to each of the daughter cells, till finally the cell has completely divided and we have a complete complement of chromosomes in each of these cells. The beautiful separation of the chromosome pairs which we just saw is tremendously important, for it has been found that the hereditary material of the cell, that is the material which contains the substance controlling the form and function of the whole cell, appears to be contained entirely in the chromosome. If this chromosome then is such a tremendously important body, what else do we know about its nature? First, it has been discovered that the chromosome is made of subunits, such as this. Furthermore, it has been found that the function of the chromosome is often very much related to specific areas of its structure. In other words, whether or not you have brown eyes or blue eyes might depend to a great extent on a particular area in a particular chromosome. We have also found out much about the chemistry of the chromosome, and two main substances have appeared from the chemical analyses of the chromosome. These substances are protein and deoxyribonucleic acid. We are all familiar with protein as it occurs in the form of lean meat. And deoxyribonucleic acid, as the name suggests, means that we have an acid substance found in the nuclei of cells. The deoxyribose is simply a sugar derivative, which is part of this complicated organic molecule. Ribose, lacking one oxygen, is called deoxyribose. The whole thing, deoxyribonucleic acid, is usually abbreviated as simply DNA. Chemists and physicists have also found out much about the structure of DNA. And some of the things they have found are represented in this model, which we have here, showing about 1/100th of the chain length of DNA. I think it is clear to you how this molecule is built up of a double strand having a spiral shape. This is about as far as we can go with the chromosomes from the chemical standpoint. And this brings us back then to viruses. A number of years ago, Dr. Stanley and others made the startling discovery that viruses, just like the hereditary material of the cells, consist of protein and nucleic acid. Viruses and chromosomes have another similarity. Both of these things duplicate. We don't know the details of the duplication phenomenon for all viruses, but we know much about the duplication of a certain germ virus. Let's take a look at this process of infection as it occurs with the bacterial germ virus. Here we have the susceptible cell, and then the virus particle is approaching. Soon it becomes attached to the surface of the cell, and shortly the DNA is injected into the cell, leaving an empty protein coat on the outside. For a while, this fibrous strand of nucleic acid exists in the cell, and we have a sort of eclipse period about which little is known. Then, after about 10 minutes, the DNA molecule divides, and then the new strands divide also, and pretty soon we have lots of DNA fibers. Shortly, the protein is built around the DNA, and soon we have complete virus particles appearing in the cell. This continues until we have several hundred such new virus particles, and then the cell bursts, releasing them into the medium. Another striking similarity between viruses and chromosomes is that they both undergo mutation. Let me explain this a bit. Suppose a chromosome were to be damaged. Often this results in the death of the cell. However, if the change is smaller, perhaps it merely results in a changed characteristic of the cell. And then, if the cell divides normally, this changed characteristic is transferred to the new cell. In other words, it's inherited. And we call this process a mutation. Now, it would be nice to study the process of chromosome mutation chemically, but there has proved to be too many difficulties to do this. So we thought, why not study the chemistry of virus mutation? For viruses also mutate. To do this, we selected the common strain of tobacco mosaic virus and then a mutated strain obtained from this common strain. Next, we inoculated a series of tobacco plants with each of these two virus strains. This is what a normal tobacco plant looks like. And when the plants like these were infected with each of the two strains of virus, we obtained the symptoms such as you see shown in this picture. This for common tobacco mosaic virus, or TMV, and this for the mutated strain, which we call HR. The next part of our study was to isolate and purify the viruses from these diseased plants, and then to examine the individual virus particles to see if they might differ in any way in size and shape. Electron micrograph pictures of the viruses showed that in both cases, TMV and HR, the virus particles seem to be identical. Further chemical and physical studies on these two strains of virus also showed a very great similarity between them. Perhaps I can explain this a little better with the use of this model of tobacco mosaic virus, which we have here. It was found that both TMV and the mutant strain HR had the sort of structure which we see modeled here. In other words, we have a section of the individual virus particle magnified many times. We see many protein subunits, and then within the structure, a single strand of nucleic acid, which in this case is ribonucleic acid, called RNA. This is slightly different from the DNA of chromosomes about which we have been talking. However, some viruses contain RNA and some contain DNA. These are very similar molecules, actually. The next step is to separate the protein from the nucleic acid of the virus, and then to break up the protein into smaller pieces for analysis. This actually represents a coiled up state of the protein, and if we were to pull out the protein chain into its extended form, we would find that we had something such as represented here in this model of a single protein chain. Although, of course, the whole thing would be very much longer than this. This is simply a small portion of such an extended protein chain. Likewise, if we take the coiled up protein from our second virus strain and pull it out into the extended form, we would have another such chain of protein. Now, in order to find out if these two protein chains are the same or different, we have to break them up in some way. And this we do by treating them with a digestive enzyme, trypsin, which is found in the digestive tract of human beings. When this is done, the protein chain breaks into fragments in this manner. And likewise, the second chain would break into fragments. If these two protein chains are the same, then obviously we would end up with pieces which are alike. And if they are partly alike and partly different, well, then we will end up with some pieces which are alike, as we have here, and some pieces which are different, such as these two fragments. The next problem is to separate these fragments of protein and to find out whether or not we really do have different pieces or the same or both. This we do by using electricity and a process which we call chromatography. Let me show you how this works. First, we apply a portion of the broken up protein to this spot on a special piece of paper. And then we subject the paper to an electric current. And we find that some of the fragments of protein will move in one direction and some will move in the opposite direction, giving us a partial separation of the protein pieces. This is not good enough, however, and so the next step is the part we call chromatography and consists of running an organic solvent down the sheet, which gives us a further separation of the protein fragments. As you can see, new spots are showing up here. Finally, when we have carried this process to completion, all of the separate pieces or fragments are spread out on the paper in a form that we call a map. Now this happens to represent a map for the HR strain, which we talked about a while ago. And here we have a similar map for the TMV strain. First of all, you will notice that there are some similar spots on both maps, such as that one and that one. However, much more exciting is the discovery of different spots. For example, notice only a single spot in this area for TMV, whereas we have a whole cluster of spots in the comparable area on the map for HR. Clearly we have here a difference in the chemistry of these two strains of virus, which will match the difference in their properties on plants. The same techniques have also been applied to the nucleic acid components of these viruses. And again, significant differences have been found. These differences appear to be really the vital differences between the strains, because it has been found that bare nucleic acid is able to cause infections in plants, whereas the protein cannot. Therefore, we think that the nucleic acid of tobacco mosaic virus is the hereditary material of this disease agent. How much of these findings will apply to the more complicated chromosome of the cell, like our model here? We really can't answer that question yet, but there are tantalizing similarities between the subunit of the chromosome, which contains nucleic acid and protein, and a single particle of a virus like tobacco mosaic virus, which also has nucleic acid and protein. Other similarities between chromosomes and viruses are that both are duplicated exactly and that both mutate. Certainly, chemical studies on viruses ought to provide new approaches to the understanding of heredity and life.
Virus was produced for the National Educational Television and Radio Center by KQED San Francisco, in cooperation with the Virus Laboratory of the University of California at Berkeley. Featured in this program was Dr. C. Arthur Knight, Professor of Virology. This series was made possible by a grant from the National Science Foundation.